So we left off in the last part with fertilization, right? So we've united the sperm with the oocyte. Their two respective nuclei have merged and fused, and now we have our first diploid cell. And remember, we call that a zygote, right? So that zygote diploid, it's 2N, meaning it has 46 chromosomes. It has 23 pairs, right? And so what's happening here is now that we have our diploid cell, we are essentially doubling the number of cells. Okay, we're doubling the number of cells without necessarily increasing the size of this kind of cell mass that we're forming, right? So this is simple mitosis. Each cell that is produced is genetically the same. It's genetically identical to the cell from which it came from. So we go from 1 to 2, 2 to 4, 4 to 8, 8 to 16. By the time we get to 16, we call this a morula. More on the morula here in just a second. But notice here that as we double the number of cells, the size of the cell, or not the size of the cell, but the size of this entire structure is staying the same. Right? So it's this compaction. We're doubling cells, but the entire size of the structure is remaining the same. So the individual cells are getting essentially progressively smaller. They're becoming more and more compact as we continue to duplicate and double these cell types, right? So again, by the time you get to the 16 cell stage, you call this a morula. Okay, a morula. So again, we're dividing. When you get to the morula, the term morula means mulberry. Okay, mulberry, and I'll show you a picture of that here in just a second. What you'll begin to have is, once you get the morula, you'll begin to have a space open up. So an opening, this kind of fluid-filled cavity, will begin to form on the inside and kind of in and between many of these cells that make up the morula. And when you see that opening is called a blastocele, or sometimes it's called a blastocyst cavity. So blastocele, blastocyst cavity, same thing. When you see that, you change the name from morula to blastocyst. Okay, blastocyst. This is this grouping of cells that have emerged from the duplication of the zygote, right? So 1 to 2 to 4 to 8 to 16 and so on, right? What you'll then have is that blastocyst, there will be an aggregation of cells kind of on one pole, on one end. Okay, so what this is called is the embryoblast. Okay, so again, you'll have a fluid-filled cavity form up. Remember, we called that the blastocyst cavity or the blastocele. So that's all of this space in here, right? And then there will be a collection, this kind of grouping of cells called the embryoblast, right? Now, you may also see the term inner cell mass. ICM, the inner cell mass. That's another name for the embryoblast, right? But notably, this embryoblast, this is giving rise to ultimately all of our other cells, all of our other tissues in our body, right? Through a process called gastrulation. And we'll talk about that here in just a little bit, right? So because it can give rise to all these other different types of cell lines and that ultimately turn into tissues and organs, these cells are called pluripotent. So they can turn into and develop into a myriad of different cell types. Okay, so embryoblast is where we're at, right? So if I go back to this morula, I said that it means mulberry. So here's an imaging of a morula. So at the 16 cell stage, and again it means mulberry. So morula is Latin for mulberry. So whoever was the early embryologist that described this, apparently they liked mulberries for some reason. So here you can kind of see a progression through essentially what we've talked about up to this point, right? So fertilization. So again, our two gametes, sperm and egg. We have fertilization. We have our zygote, the uniting of our two nuclei. So two haploid nuclei produce our first diploid cell. We go to 2 to 4 to 8 to 16. 16, we call that a morula. We form that blastocele, right? And we'll talk about what happens in this blastocele in a little bit, what happens with this embryoblast or this inner cell mass. And then ultimately, this is going to implant into our uterus. Okay. Now, all of this that's occurred thus far, so all of the duplication from 2 to 4 to 8 to 16, all of this is occurring inside the fallopian tube. Okay, this is all occurring in the fallopian tube through the first several days, right, after fertilization. So it's occurring in the fallopian tube. So you can take this to mean that the size of this collection of cells, it can't be getting extremely large because it's still in a very small tube, right? It's still in the fallopian tube. So notice here that our zona pellucida, right, this layer surrounding the oocyte, it is still intact, right? It's still present. So it's present there, it's present here, it's present here, it's present around the morula. That's helping to keep the overall size of this structure the same, right? It's helping to keep it relatively small, right? And then ultimately, once we get near the uterine cavity, the cell mass, right, this morula will, and this blastocyst is ultimately going to burst out of that zona pellucida through a process called hatching. Okay, so we actually hatch, right? We hatch. We don't hatch through an egg, but we hatch out of this glycoprotein zona pellucida, right? It probably feels really good whenever you finally hatch out of it, and now the blastocyst is able to be free, right? It probably feels good. So once we have made it to the uterus, now that blastocyst with that embryoblast, right, that inner cell mass, needs to insert itself into the uterus, right? Because this is ultimately where the embryo and the fetus is going to develop, right? So normally implantation is in kind of the posterior wall of the uterus. This is typically where implantation occurs by the end of the first week, right? So you can kind of see a general timeline here. Day 6, day 7, day 8 or so, you're beginning to implant, right? So that zona pellucida has broken down. So you insert the blastocyst into the uterine cavity. That inner lining of the uterine cavity is called the endometrium. You may recall there's 2 layers to the endometrium. One is called the stratum basalis and one is called the stratum functionalis. So notice here where this term says functional layer of endometrium, that's the stratum functionalis. That's where the actual blastocyst is going to implant. The functional layer is going to be sloughed off if there's not fertilization, there's no implantation, and lost in menstruation. And then that basalis, that stratum basalis, is essentially going to grow and replicate and divide and replace the functional layer, right? But for our purposes, think blastocyst is implanting within that stratum functionalis layer of the endometrium, right? Now, once this happens, there's a differentiation of cell types, right? So we've already mentioned this embryoblast before, right? This embryoblast is surrounded by another layer of cells called trophoblast cells, right? And some of these trophoblast cells, these are what are ultimately going to give rise to the placenta, right? To our placenta, which is the site of nutrient gas exchange, waste exchange at the between the mother and the fetus, right? So there's 2 layers. The outer layer is called a syncytio trophoblast, and the inner layer is called a cytotrophoblast. That term syncytio means like a big functional unit, right? So you can kind of see here all of these orange cells, kind of all of these here, all of these here. Hopefully you can see my cursor. That's the syncytio trophoblast. They kind of merge together and form this one really large structure, okay? And then these cytotrophoblasts are these that are closer to the embryoblast, right? Now, we have implanted into the uterus, so we had to maintain that uterine lining, and so you need to ensure that that uterine lining is going to be intact, right? So if you think about our process of folliculogenesis that we've talked about previously, right? So folliculogenesis, you are developing the follicle, you ovulate the secondary oocyte somewhere around day 14, and then you form that corpus luteum, right? So it was the Graafian follicle that released the oocyte, and then that turns into the corpus luteum. The corpus luteum, remember, it was producing primarily progesterone. Okay, it was producing primarily progesterone, and that helped to maintain the uterine lining. Okay. Now there's another product. There's another hormone that's produced called HCG, this human chorionic gonadotropin, and this is produced by the blastocyst, right? So by that implanted collection of cells, and this is going to help to maintain the corpus luteum such that it continues to produce progesterone such that you keep the uterine lining thick, vascular, and you prevent menstruation. So if you recall, if we didn't have fertilization, that corpus luteum would turn into the corpus albicans. The corpus albicans is non-functional. Progesterone levels go down. Menstruation occurs, right? You lose that uterine lining. Well, in this case, we don't want that. We don't want to lose the uterine lining because we've just implanted a blastocyst there, right? So this human chorionic gonadotropin from the blastocyst helps to maintain and keep the corpus luteum, so you continue producing progesterone so that you maintain that thick stratum functionalis layer of the endometrium, right? This is a hormone, HCG, so it is broken down, it's lost in the urine, and this is the basis of pregnancy tests. Most pregnancy tests are trying to detect and determine whether or not there is HCG in the urine. Right. Now, after a number of months, usually the first couple of months, then the placenta will take over. Right. The placenta will take over. It will continue producing estrogen and progesterone and maintain that thick endometrial lining to serve as a place for the embryo and then ultimately the fetus to develop, right? So let's stop here. We'll pick up in our next part and we'll take a closer look at that embryoblast, right? So we'll take a closer look at this inner cell mass, these embryoblast cells, and we'll begin to see what's actually going to happen to those and how do those ultimately turn into all of our different cells and tissues.